Okay, so is everybody looking at the Pivot Interactives interface right now? Everybody can see it? Okay. So this is what the new activity is going to look like. And you can see the video is the same as the last one because you're using the same interface to take the data, okay? So basically, you have options. You have enough room here to take and analyze data for 3 different experiments because there's 3 different things that we're considering as independent variables that could affect the amount of force required to keep the cylinder in a circle, right? And so I'm going to ask everybody to take data for 1 experiment minimum, but then you can go on to do more if you have time and if it's interesting. I think if you don't have too much trouble taking data, then it won't be much harder to take data for 3 experiments instead of 1, and then you could actually have all the results for yourself, and that would be kind of cool. But your mileage may vary. So your first thing is to state in this little box, what quantity did you vary, what quantities did you hold constant, and what are you trying to measure as your dependent variable. For everybody, the dependent variable is always going to be the amount of force. But I want people to actually record it there because that'll help me know whether people are basically on track or way off. So write down your whole experimental design in this space, okay? Just recording. My students are in breakout rooms right now, but they're working on a common problem set. I just wanted to show you what that looks like. If I pop over here to, all right, so I'm hoping this is now recording the Google Slides window. So I can look over here in the sidebar on the left side and see which students are working on their slides. Right now I've got them all working on the same problem. They're drawing diagrams the same way we normally would. I was able to pop into this group and ask them to add a kind of diagram. So when I saw that they had done this work and I wanted to see an extra vector addition diagram to show where they got 225, I was able to hop over to their breakout room and ask them to make this additional picture, which they're making now, which is kind of cool. So we started out by reviewing student work on this previous problem, and then I asked them all to work on this similar problem and try to figure out that way. See, so here these guys have completed this. And now before the end of class, we'll be able to come back together. And since everybody can see all of the slides, we can kind of have, this is sort of similar to when we use our whiteboards in regular physical class. And now we can kind of look at if we all have a consensus on how this is done.
And then the last thing that I want to go over is how we test for blood type. So first, when testing for someone's blood type, you take a sample. Then you mix the sample with anti-serums and observe what happens. Now, if we were in the classroom like I had hoped we would actually be testing, it wouldn't be real blood, they'd be fake blood, but we'd be actually doing this ourselves in a petri dish and all of that kind of stuff. But we're going to be doing it virtually through this experience. But when you mix the sample with anti-serums, and you get to observe what happens, and that tells you what the blood type is. The serums that you use are anti-A serum, anti-B serum, and anti-RH serum. So anti-A and anti-B, that sounds a lot like the antibodies that we use to test for blood type. All right, so when we use these anti-A, anti-B, and anti-RH serums, we then get to see what happens. Now, specifically what happens is something called agglutination. So agglutination is a really fancy word that just means the blood clumps. And if clumping occurs, it means that the red blood cells have those antigens on the blood cell surface. So if we look at these 2 pictures, in this top one, all 3 of these vials experienced agglutination, meaning that the blood cells clumped together, leaving behind this almost like yellowish clear fluid around it. So what that means is that this red blood cell has the A antigen because we clumped here, it has the B antigen because it clumped in B also, and it has an RH antigen. So someone with this response to their blood type is actually A, B, and then because this also agglutinated is AB positive. Now if you look down at the bottom picture, A here, well, we see it didn't clump at all. It's still bright red. Same with B, it didn't clump at all, but we can see agglutination here in our RH. So if it doesn't clump in A, that means it doesn't have A antibodies, or an A antigen, I should say. If it doesn't clump in B, it doesn't have B antigens either. So that means that it's type O, because only type O doesn't have any antigens. And we can actually go back to this particular picture that we were looking at before, see, antigens in red blood cell, none. So there's nothing around it at all. So it would be O and then Rh positive because there was clumping. So whenever we see clumping, whenever we see agglutination, that means that it has that particular antigen. And that's how we tell what someone's blood type is.
Like I was looking at the World Wildlife Fund, and they're trying to eliminate bycatch and do like turtle-friendly hooks when fishing. We can also protect their habitat, so obviously recycling, don't leave your trash on the beach, things like that. And they also satellite track the sea turtles so that they can see where like their nesting beaches is and try to protect the area, and then create like protective laws, federal laws and other state laws to protect the sea turtles.
Interesting.
Yeah.
Okay. I forget where I saw it, but I do remember seeing in the last couple of days, and I think probably a lot of you have heard this, how there's been a huge uptick in the amount of wildlife that's just out and about because there's so few people around, and they've noticed markedly an increase in the number of sea turtles coming to particular beaches now to nest where they haven't seen any turtles for who knows how long.
Right.
So it's just kind of interesting, and just as sort of a side note, you know, I think it's going to be very interesting to see what lessons we learn from this whole experience. And, you know, maybe there are certain changes in our actions that we might consider keeping, because it's kind of hard to deny we're seeing many improvements in the environment just in this last month from the reduction in our activity. So obviously we can't stop everything we do, but I wonder if any of our behaviors might be continued in order to promote the health and well-being of some of these species that we're losing.
As I said at the end of the last lesson, this is probably the most complicated piece of everything that we're talking about with controlling gene expression. There's one mechanism, complicated about one mechanism. Well, there's a lot of parts. One mechanism here, and it's known as an operon. Operons or operon systems, depending on you know how you phrase it, are unique to prokaryotes, and that's a really, really important point. There have been multiple occasions, either on class tests or in a few cases on the exam, where students have been asked to explain a eukaryotic mechanism, and they've explained operons, which means 0 points. Hopefully that is not going to happen this time around. All right, unique to prokaryotes. So what an operon does, it basically can shut down or turn on an entire biochemical pathway by simultaneously turning off all the genes or simultaneously turning on all the genes. That is something that our cells, eukaryotic cells, cannot do. All right. Now, why is it that prokaryotes can do this? They've got aspects of molecular biology that we don't. All right. First off, unlike in eukaryotes, all the genes for a particular pathway can be right next to each other on a chromosome. Bacteria have one chromosome, right? They have one circular chromosome. We have many linear chromosomes. So take a pathway with 3 genes on it. In bacteria, if those 3 genes, you know, they're coding for enzymes 1, 2, and 3 in the pathway, they could be sitting right next to each other, literally, on that chromosome. In our cells, if we had the same gene, which would be doubtful, but if we did, enzyme 1 might be coded for on chromosome 5. Enzyme 2 might be coded for on chromosome 18. Enzyme 3 might be coded for on, I don't know, chromosome 11. There's no way you can turn them all on or off at the same time. You'd have to regulate each one individually. All right? That's one thing that bacteria can do that we can't.